Our next speaker is Henner Koch from Aachen, Germany, and he will take a network perspective on structured events in the human cortex. Take it away, Henner.
Yeah, thank you very much, Hype, for the introduction. Yeah, thank you. And also, I would like to thank the organizers for organizing this meeting. It has been very pleasant, as has been all the other human tissue meetings I have been to. And today, I would like to talk about network events in the human cortex and how they are structured, meaning that they have components that we can detect over a big sample size, but also that there is a diversity in these network events. So I would like to show you a little bit background. Of course, we know that the human brain and other brains as well are very good at producing rhythmic activity. And you can see that, for example, in the brainstem and the circuits that generate the rhythm that lets you breathe. It's a very important rhythm, of course, because without it, you're dying. Then, of course, there are other rhythms, for example, in the spinal cord that let you walk. And also, there are rhythmic activities in the cortex, and this is shown here. So on the top, you have an EEG of a patient that is awake and relaxed. So you have alpha rhythms, and then you have the same patient during non-REM sleep. So there you have higher amplitude theta waves, and so it's a switch of rhythmic activities from one state to another state. So in my talk, I would like to answer somehow, are there mechanisms that we can detect that govern rhythm-generating principles in the human cortex? And more specifically and scientifically, I would like to ask, can we detect principles in physiological and pathological network dynamics using MEA technology? And how large is the diversity between slices from the same patient and between the patients? And also, do we detect any influence of age, gender, and pathology of on these network events? So in Aachen, our team, of course, consists of a big clinical team. So we have neurosurgeons that provide us with the tissue, but we also have the EEG teams to generate epilepsy cases. So we take both tissue, excess tissue from epilepsy and tumor cases. If there is a decision for surgery, of course, only if the patient consents to it, we will then take the tissue for research, as shown here. So the neurosurgeon is carefully resecting a part of the cortex to get to the pathology. And then we can use this tissue, as you have heard before in many other studies, to, for example, study physiology. And I hope you had time to go to the posters of Dr. Xi, Dr. Yang, and Dr. Bach that have shown some nice patch clamp results, but I will not talk about patch clamp today. And we can also investigate the pathology activity, for example, investigating how the hippocampus is producing network activity, but I will also not talk about hippocampus today. So in the cohort we analyzed, it was 23 patients. We had, as I said, tumor excess tissue and epilepsy excess tissue. We took slices from frontal, temporal, and parietal cortex, investigated these. So as you all know, brain slices can be then prepared from this excess tissue. And in Aachen, we focus a lot on human organoid brain slice cultures, as is shown here. So what we found is that if you use a human CSF as a culture medium, you have the best success for giving long-term cultures. And as Jonathan nicely showed, you can use viruses to look at your samples, how viable they are. And then you can do recordings. And these are a little bit different recordings to the patch clamp recording that we've seen before. So here you see a MEA chip, and on this MEA chip you can put your slice. You have a recording chamber, similar to a patch clamp recording chamber. We have a perfusion, control temperature, and all these things. And then you can record activity patterns on these chips. And I will show a little bit more technically how this is done. So here, again, this is our chips. We have a 16 times 16 electrode grid, which is spacing of 200 micrometers. The electrode sizes are 30 micrometers, and that gives you a pretty broad recording field. So here we have a cortical slice culture. You have AAV-induced expression of GFP, and then you can also map all your electrodes to the distinct layers. And this will be more important later. But also you have to keep in mind for the MEA recordings, we will record from this electrodes, the activity from the surrounding cells, so you don't have really knowledge about what cell produced the action potential that you're recording. And a typical recording is looking like this. So here you have the voltage, you have your action potential that can be detected, and can extract the waveforms and analyze them if you want. And what's important is you have pretty high temporal resolution, but only mediocre spatial resolution with a 200 micrometer distance electrodes. And this is a little video of a typical recording in such a slice culture, how it works. Yes, so you can see there's this sparsity, and every now and then there's also these synchronized bigger events. Supposed to be also sound, but it didn't work apparently. Anyways, and to detect these network events, you can basically easily just calculate the population firing rate. It is just taking the number of spikes in a certain time bin over all the electrodes, and it will give you a curve like this. So this blue trace is a population firing over all the channels. You can see at some point there is such a network event. You get a bit increase, and it crosses your threshold. So then you can set this as the onset of your event, and then also you have an offset of the event. That, of course, would allow you to calculate the duration of such an event. And if you record them over time, as I showed you in the video, they come repetitively. You can then also calculate the inter-event interval of these events. And if you plot then the whole cohort of many recordings, as it's done here, so you can see on average, these events are rather short, so they are shorter than 300 milliseconds in duration. But they have a pretty long tail to the longer durations. And also you have pretty low frequency, so they come around every 10 seconds on average, but they can become also much more long in their event interval. So, and of course you can manipulate your system as is shown here. So under baseline condition, you have this very low frequency events, but then if you increase excitability in your system, with high potassium and GABA-zine, you get a lot more frequent events without also a little bit change in duration. But as you can see here, when we plot all these different experiments, the green dots are the baseline events, and then the other ones are the ones where we increase the excitability. And you see there's much more increase in the frequency, which we call the downstate duration, getting slower. Then there is an increase in the upstate activity. So it means most of the structure of these events seems to be preserved with high excitability. And if you then look at the spatial distribution of these events, as is shown here, in ACSF, you have a very predominant activity pattern that comes from the deep layers, so layer 5 and 6. And then with high potassium GABA-zine, it changes a little bit, so you still have the same core of cells that fire during the events, but you also activate layer 2 and 3, and this is again showing the same thing over the whole cohort. So in a baseline condition, layer 5 is really dominating all the other layers, so the main activity comes from that layer. And when you go up with excitability, then layer 2, 3 can also become similarly active in your slice. So this brings me back to one of my hypothesis when I was a student. So are there some cells that drive network activity? So in the lab I studied, the pacemakers were a very important concept. And to study this, we look at these onset time of the events and look if these always the same cells are leading these events, even a little bit before the start of the onset, or within the first couple of 100 seconds of the event. And we kind of divide them in these pre-onset cells and then post-onset cells, and the red ones are the first that come in the event. And if you then plot the spatial distribution of this leading and initiating cells or electrodes, they are again dominated by layer 5 neurons, and this is also shown here. So really the pre-onset, very early cells, and also the core cells are all located in layer 5, so it really seems to initiate these up and down stand events. And then we wanted to know how reliably is this done between different events. And for this we used the Jacket score. So what it does is you have two sets of events, and then you just look how many of the features are overlapping. So you can see these circles here are 2 events, and then these are the same initiating neurons in these events. So then you have a certain amount of overlap, and that is calculated by this score. It goes between 1 and 0, and you can see here these are plotted for subsequent events, so it's always a pair, and you can see the mean is pretty low, so only 15% of the initiating cells are the same in all these pairs. And this is again shown here in one example. So if you look here at these different events, they look quite similar if you look at the overall firing rate of the events. But if you look at the raster plots, you see that they are very different. You can see the green, blue, and orange are 3 different events that are overlapped. And again, we calculated, for example, for this score, and you can see it stays at this very low probability, and that holds true for the whole cohort. So if we do that over all the recordings we've done from all these patients, the median of the Jagged score is 0.13, so pretty low. And the activation entropy, another measurement for the same thing, is quite high. So in the end, what we know is that the real pattern of spikes in such event is pretty stochastic. But what we also see, and this is a quantification for the whole cohort, is layer 5 is the place where all these events are getting initiated. And the last thing I wanted to look at was propagation. So again, we have layer 5 as an initiating zone, and then you have starting propagation into the upper layers, and they get then terminated often also again in layer 5. And of course, it is nice to see that this holds true to some very old data from the ferret. And we have here in the audience the author of this paper, where she also showed that in this up and down state in the ferret, you have initiating and driving in layer 5, 6. And this poor poster that fell down showed the same thing for acute human slices as well. So it seems to be a very conserved concept. And the last thing I looked at was the velocity of propagation in these slices. And this is done with the center of gravity method of the spikes propagation through the slice. And you can see we get the velocity of all these different points, and then you get an average value. And again, we plotted this for the whole population and get values that are pretty close to what is known for cortical circuits for propagation speeds of around 300 millimeters per second. So one of the questions I asked in the beginning, is there any influence on the samples on this activity? And one I plotted here. So I didn't find anything between epilepsy patient and tumor patients per se, but we found the older the patient was, that the activity was less pronounced. So there was less active channels in the MEA recordings. The mean firing rate was lower, and surprisingly, the number of events was not much lower. So it seems that the networks are less strong, but they're still there. And the other effect I looked at, it was just the culture, of course, because we worked all these out in these human organoid brain slice cultures. And you see there is a little downward correlation to the days in vitro and the firing rate, for example. But you get still a lot of these events also in a long time culture. So it seems to be pretty stable and also nicely comparable to the acute slices. Okay, this brings me to my conclusion slide. So I showed you hopefully that we have these network events that are really driven by layer 5 and 6, and that also the initiation zone is in layer 5 and 6, but that the real fine details of this initiation is pretty stochastic. So it's not pacemaker driven like we hypothesized some time ago when I was a student. So with this, I would like to thank everybody that helped me. For, of course, everybody in the lab. Aniela has now moved out of my lab into our colleagues' collaborator lab in Jülich. And then, of course, always talking to Hyde and Natalia about the newest experiments that are possible. And with this, I thank you and are open for some questions if you want.
Thank you very much, Henner. An exciting talk.
Yeah.
First question over there, then there, and then there.
Thank you, wonderful talk. I have a few questions. But maybe, okay, so let's start. First of all, I'm wondering whether, did you do any clustering on your data? So did you differentiate between the different action potentials on your recording data? Because then in that way you could say whether those are excitatory, inhibitory cells, bursting cells or not. So that's one thing. My other question, whether could you comment on that normally in the human brain and also in other animals, upstates and downstates are slightly different than what you see and what we also see in the neocortex. So during upstates, there are lots of firing, but during downstates, there is nothing, basically. No one is firing, no neurons. But in these preparations, we still see several cells still firing. So I'm not really sure that this is real slow oscillation, which could be related to real, human sleep or human slow oscillation.
Sure. I mean, it's a slice, and what you see is, of course, that during the downstate periods, if you want to call it like this, it's pretty sparse. So there will be some random activity in the slice. But if you would record from one cell, somewhere in the slice might be that the cell not doing anything. So, but during the upstates, you get a pretty much strong synchronization of cells. So I think. Not 100% sure if there is no activity at all during the downstates in vivo. I don't have done any in vivo measurements, and yeah, I also don't know if up and downside are the perfect definition for this. What we see are recurrent network events that seem to be stochastic. They are at the frequencies that we see also in the ferret or mouse, I guess. So there seems to be like an inherent pattern of the cortex to generate this kind of activity, maybe as a resting state, but I don't know. And for your first question, we try to cluster them and also try to look at can we make inhibitory versus excitatory clusters like Buzaki and so forth. But in my efforts, it was always very low interneuron participation, and it could be because at the moment we're using a threshold method to detect the spikes, and if the action potential of inhibitory neurons might be smaller, we miss a lot of those. So maybe I would have to use a different detection method. Yeah.
Okay, question over here.
Thank you so much for the talk, and this is pretty exciting that you got that to work.
Okay,
so actually, my question is the same pattern that you see. It's also seen in two-dimensional networks and organoids derived from iPSCs. It's called the network burst. That's what you're showing at network burst. I think that's what it looks like. Of course, when you do a slice, the same thing when you do an iPSC-derived preparation, there is no external inputs, unless that you just input with electrodes. But the cortex is pretty desynchronized when you go in a state. In the normal, if you record extracellularly, the cortex doesn't look like having those events. Probably people will call it epilepsy on a dish or something like that. Some people have told me that. My question is, do you think that this event that you see is something that is intrinsic to this network, and networks that don't have an external input, and they have to go into some sort of internal mode, that they go into this up-down state network bursts and they go, and they're definitely informative, as you show with age. But do you think that this is what is happening here, that the network is just taking over, and now this is my dynamic because I'm not talking to anyone, so they go into some kind of internal loop?
Yeah, it's very difficult to answer that because we don't know. What we know is we see it in a lot of these slices at different conditions. So I didn't do acute MEA measurements because acute MEA measurements don't work that well. For some reason I also don't understand, but it's more difficult. But I think that these are the inherent activity patterns that just arise if you leave them isolated, let's say. Of course, if you have your slice back in the brain, then it will be dominated by the inputs and outputs of the system. But now it's isolated, and this seems to be like in that small network that's left is to be the intrinsic pattern that we generate then, I think.
Thank you.
Very, very nice talk. Thank you.
Yeah.
Did you try, you mentioned that, and I've seen the poster, single cell recording, patch clamp recording. Do you see the same activity? And do you have any way, I'm thinking of the method proposed many years ago by Yves Regnac, to understand by different in voltage clamp, by different clamp holding potential.
Yeah.
What is the initiation and what is the inhibition and excitation? That's it.
So I haven't done any recordings at the moment where I have a MEA and patch clamp.
Without the MEA.
Without the MEA, with just one extracellular electrode, we did some of these.
Intracellular.
Yeah, we did the combination of intracellular and extracellular recordings, so that works, and they correlate. But I think I would like to do as a next step to really record single cells on a MEA, because I think it will give you really the answer. Yeah. And for this, I can go back to blind patching, like I did when I was a student, because I don't, on the MEA, you cannot put it, it's very difficult to also have it on a big microscope for like visual patch clamping, so this would be the idea, yeah.
Watch out about the ground. It's tricky, MEA and patch clamp, it's a nightmare.
Okay, well.
We take 2 more questions. First Albert and then Mavi.
Well, thank you. It's a bit speculative, but maybe you have the intuitions to answer this. And I'd like you to link to Natalia's talk and to ask you whether, if you'll get slices from high IQ versus low IQ people, what will be the kind of differences in the network response?
Well, that's a very nice question. I don't exactly know. It would be very interesting to measure. I mean, I could imagine that
I'll just continue. The question is, so you did patch those cells, some of those cells at least, and the differences in the physiology of the high versus low IQ are known, right? They measure that. So would you care to speculate on that?
Well, I find it hard to I don't know what will happen to the network events if they change to this lower input cells. Maybe if you put it in a simulation that is not only simulating single cells but simulating the network, then that we can answer that maybe better. But without, I will be just speculating in the wild. I don't know what will happen. Do you have an idea what could happen?
Well, I was hoping maybe that the simulations output, and you'll see some jitter or even high responsiveness depending on the level of potassium and GABA.
Yeah, interesting.
Great point. Final question for Mavi.
Hi, thanks for the talk. I was wondering, you have very high propagation speeds. So are those in the control condition or in GABA zinc with high potassium?
The propagation speed was
200 and...
Yeah, they were calculated in ACSF conditions. They go up a little bit higher when you block inhibition to 400 or so. Yeah.
Yeah, but they are quite high.
Quite high, yeah.
Yeah.
Well, that's what we measured.
All right, with that, let's thank all the speakers of the session.